All right, outstanding. Thank you so much for inviting me here, and it's, I believe, a great designation to be the last speaker in the summit. I guess I'm the only one holding you up here. So I'll try to make it a little bit, I wouldn't say fun. I mean, science is fun for me, but I don't know how much fun it's going to be for you to hear what I feel is fun. Anyway, so when you look at this presentation, the title is pretty long, so that kind of explains everything that we're going to talk about. So my research group, I'm here. My research group works on, we are mostly focused on respiratory diseases, so we work on developing therapies that could be used to either repurpose a drug for diseases that occur in the lungs. That could include lung cancer, could include a rare cancer that we work on, mesothelioma, could work on pulmonary infections associated with cystic fibrosis, very common, very deadly. We also work on a couple of rare diseases, pulmonary arterial hypertension and idiopathic pulmonary fibrosis. What you're going to see today is the results from a project that was pursued by a master's student, and then we completed the study with the help of the student. As you will see through the presentation, you will see that this project follows an academic timeline, right? So with academic timeline, I mean our experiments today are defined by the results from yesterday. So we are not necessarily following a set project plan. We are always looking to improve how we move forward. That's number one. The other disclosure I have to make today is that I'm not claiming to be an expert in anything, right? All right, so I'm telling you a story that my students developed with an idea I had. So I'll be happy to answer your questions, but I'm here to learn as well from expertise. Brief introduction about me. I'm an associate professor of pharmaceutical sciences at St. John's University College of Pharmacy. St. John's University, if you're not aware, has been around for 150 years. We have a very large graduate master's and PhD program in pharmaceutical sciences, about more than 100 students doing research, about 40 active research labs on campus, extensive NIH funding, industry collaboration. My research group works on multiple things. So the first one we work on is drug repurposing, where we are looking to develop new therapeutic indications for a drug that has already been approved by the FDA, or at least has entered clinical trials for a different indication. The other thing we look for is developing novel nanomedicine-based approaches for localized lung delivery. And something that is very dear to my heart is developing scalable nanoformulation. I have been doing work in nanotechnology, nanomedicine since the first day of my PhD. And as an academic, now that I run my lab for last almost 10 years, I feel that the only way forward for academics or professors is to take a system and make it more and more complex. So we take a nanoparticle that could be made maybe in 2 steps. Like, oh, let's add some targeting to it. Let's add one more targeting moiety. Let's make it more complex. That's the way to go to develop another or to publish another article. That is unfortunately been the case. What we want to work on is to develop systems that could work or serve the needs that are needed, but can be scaled from a lab scale, from an academic lab, where we are only worried about a couple of milligrams of formulation, all the way to kilograms. So we need simpler processes for that. So once you look at this picture, would you like your formulation development to be like this? Again, I'm not here to tell anybody that any processes that people follow that have been developed, have been commercialized are bad. Everything is there for a reason. But would you like this to be your process development, or this, where your excipients go on one side, from the other side you get a single step formulation made for you with very good polydispersity index, very nice characterizations and everything ready to go. No cleanup required. Right. I'm more inclined toward the one on the right. Right. So before we move forward in how we want to approach this problem of pharmaceutical scalability, I'll give you a little bit background about the disease that we work on. And I understand this, the audience is more pharmaceutics, drug delivery oriented, so I'll keep my introduction to the disease that this project is focused on kind of brief, but happy to answer any questions that come across. So the disease that we work on, this project was focused on, is cystic fibrosis. It's a rare disease, extremely rare. So the definition of rare disease in the United States is less than 200,000 patients in the country. The definition obviously changes based on population of each country you go to, but for US, it has been around 200,000 patients, active patients. So there are less than 1,000 patients that are diagnosed with cystic fibrosis every year. At this time, there are approximately 30 to 40,000 patients in the US living with cystic fibrosis. Most of them are diagnosed when they are babies. Cystic fibrosis is a hereditary disorder that happens because of double recessive mutation in cystic fibrosis transmembrane conductance regulator, or CFTR gene, specifically F508 del mutation. So what this protein does, this protein this gene causes, this is the pointer, maybe I can go here, develops CFTR protein that is responsible for movement of chloride ions out of the cell. What that chloride ion movement does is creates the movement of water in the body, creates a mucus. Mucociliary clearance, all of you must have heard about it. So mucociliary clearance happens because the chloride ions are being moved forward. In mutated CFTR gene in cystic fibrotic patients, this chloride movement is not happening. What that does is creates a very thick mucus that is unable to move. It affects all across the body, but the most severe effects are seen in digestive system, in pancreatic systems, and in the lungs. When the mucus is not able to move, mucus is one of our body's defense mechanisms. When the mucus is not able to move, all the foreign particles, microbes, our body is not able to clear them out. We are more susceptible to get infection. One of the main causes for fatalities with cystic fibrosis is lung infection. So if you look at this figure, this basically tells you how the cystic fibrosis happens. So there is, even with parents carrying one recessive gene for CFTR or mutated gene, there is only 1 out of 4 chances that the baby will have cystic fibrosis. Again, as I discussed in the last slide, when the airway is clear, you are able to clean out or get rid of foreign material, pathogens, etc. quickly. When the airway is not clear, it's lined by thick mucus, things get stuck. And because of that, you will get a lot of issues. In case of digestive system, the enzymes will not be able to reach to the digestive tract. You will have problems associated with nutrition. The children with this disease will be malnourished and everything else. Now, talking about lung infection. If you talk about lung infection, if you look at this figure on the left, you will see that in the normal airways, you will see the bacteria. The mucus is thin. The mucus, with its movement, is able to clean out the bacteria. But in cystic fibrotic patients, there is a thick mucus. The body is employing antibodies in PMNs to get rid of that infection, that bacteria, but it is not able to move out. It gets stuck there, and that's what causes infection. It creates biofilms, causes infection. If you look at this graph from a couple of years ago, you will see that as the patient's age increases, different bacteria are more susceptible to different infections. I am personally in today's talk focused on Pseudomonas aeruginosa because that is the most common bacteria infecting lung airways in adult population. So if you look at this figure basically tells you about inhaled, and again, there are many treatments available for cystic fibrosis, but they do not treat the lung infections, they treat the disease. They cause they change the chloride channel movement, and they change the CFTR mutation, but they do not treat the lung infection. If you look at this slide basically tells you as you progress from year 2009 to 2016, and I'll tell you why this is important. The primary antibody or primary treatment for lung infection was aminoglycosides or tobramycin, etc. About 95% treatment patients received tobramycin, at least one of those. And aztreonam, the drug that I'm interested in, was only 3.7%. In 2010, aztreonam was approved as an inhaled therapy. 3 times a day, 75 milligram in one dose. And as you will see, the fraction of patients getting aztreonam at least as a mono or a combination therapy is increasing from 3.7% all the way to 43% in year 2016. That means inhaled therapies work for lung infections. I mean, it makes sense, right? You are putting the drug where the disease is, right? So more than likely it's going to work. And at the same time, what we are also doing with inhaled therapeutics is you are protecting the rest of the body, excluding lungs from the side effects these drugs may have. All drugs have side effects at one level or the other, right? So by producing inhaled formulations, you are protecting the body and you are providing high local concentration of the drug in the lungs. Now, what are the compliance issues associated, however? So now you will ask, this aztreonam that you want to focus on, it's an antibiotic. There is an already available inhaled formulation for that. The drug does not suffer from any of the solubility issues. So what is it that you want to do, right? So you will see that the inhaled antibiotic compliance, once you start looking in literature, is around 50 to 78%. And it is very tightly related to the number of dosings patient has to take and the steps they have to take before the formulation is ready to go. In case of Estronam, marketed with the brand name Caiston, it has to be taken 3 times a day, and every time the patient has to reconstitute the dry powder provided to them with sterile saline before they can inhale it using a nebulizer. So there are multiple things that they need to do. They have to carry the nebulizer with them. Obviously, if they're going to do 3 times a day, they have to keep it handy, and they have to be they have to be willing to take the powder, put the saline, and make sure they don't make a mistake in the process, right? So can we improve the compliance by changing the stability issue associated with the drug, or by at least decreasing the dosing frequency, right? Now, one of the major reasons we have to give the drug 3 times a day is because of the very short half-life. The half-life for Estronam I believe in humans is around 2 hours, or less than 2 hours. So unless you create a controlled delivery system for that, it will not be able to sustain the therapeutic concentrations for over a day, even if you were to develop once-a-day therapy. The other problem is the pricing associated with it. So there are currently 2 marketed inhaled antibiotics for cystic fibrosis-associated infections, Caiston, Estronam being one of them, costs around $11,000 a month. Tobramycin inhaler costs around $11,000 as well. One more inhaled antibiotic was approved a couple of years ago from Insmed, Ericase, as the liposome, amikacin liposomes, also costs around $11,000, but is not closely related to cystic fibrosis infections. Now, when you talk about lung delivery, and we have these problems to address, there are, so the approaches that we need to take for oral delivery versus inhaled are slightly different because we are limited with the kind of dosage forms that we can use for inhaled therapeutics. I wouldn't inhale a tablet or a capsule, right? I mean, it has to be in some form that the lungs, that wouldn't block my lungs, or that the lungs are able to inspirate, and the body's defense system mechanism doesn't spit it out, right? So we are limited with things that we need to be sure about while developing inhaled dosage forms. So when you look at this figure basically tells you when you're developing a nanocarrier, you are not only protecting the drug from external environment, so that will potentially address the problem of of short half-life. You're also creating or providing site-specific delivery of drug to their targets. In this case, it's local delivery. We are not going after a particular region of the lungs or a particular cell or a particular receptor. We are doing local delivery. We are also looking at prevention of unwanted burst release. We are providing drug in a fashion that will slowly release the drug into the lungs, thus maintaining therapeutic concentration for a long time. Triggering drug release at the site of action, that the dosage form goes to the lungs, start releasing drug, and if possible, if needed, co-delivery of combined drugs, create a system for that. Now, all of this is great, right? Now, I want to focus on lipid nanoparticles for inhaled delivery. So the earliest class of lipid nanoparticles, liposomes, as Dr. Ali just mentioned, I think it was discovered in 1965, if I remember from your slide, been there for a while. Again, but there are issues related, as Dr. Ali mentioned, lipid bilayer, issues with drug release, compatibility kind of drugs it can carry, and capability of delivering small molecules. So lipid nanoparticles have been around for quite some time. A lot of work has been done. As I mentioned, Ericase is the liposomal formulation that's been out there. Now, as many of the speakers have discussed today, and Dr. Pradham talked about it in great detail, about scalability of lipid nanoparticles. So all of the lipid formulations you're looking at, many of them are currently prepared by batch process, that you're going batch after the batch. There is no process that exists for continuous manufacturing of these products. Scalability remains a major challenge in clinical translation. I could create the fanciest system possible in my lab with X number of ligands, antibodies, and everything else attached to it. Once it goes for translation, it may have the best result in vitro, in vivo as well. Once it goes for translation, almost every time you have to reinvent the wheel, re-optimize the formulation, redo the process design and everything else. And at the end of the day, the formulation you may get, which may be translatable, it may look nothing like that your research lab developed in the university, right? It would look something different. So wouldn't it be great if you could develop a formulation that could be scaled, could be translated the way you made it in the lab, right? A simple process. Now I'm going to talk about an equipment that all of you are familiar with, right? Except myself. I have little bit knowledge about this. Hot melt extruder, or extruder, as I was talking to somebody else. Right? It's a continuous process. It has been used extensively in pharmaceutical industry. Some universities have access to this as well. Fortunately, St. John's is one of those. The machine can be run continuously depending on its capacity, and it can produce formulations. Historically, traditionally, HME has been used to develop amorphous solid dispersions for solid dosage forms. What we started looking at is, can we create a process, optimize a process to use HME to develop lipid nanoformulations? What is it that we can do that instead of getting a solid output, can we get a liquid output, something that is in sub-nanometer range, about 200 nanometers or less, that does not require further processing, right? Or if it does, very basic minimal processing. So this slide basically tells you about where, I don't have to discuss too much about it because all of you are familiar with how HME works, is how HME process goes. So you are basically feeding the material. If you see here on the side, it goes through a set of screws. I'm showing here is a twin screw extruder going through a set of screws, multiple heating zones, and at the end of the day, at the end of the formulation, you'll get the product here. What we also see here is that there is a place in this HME to add liquid. So you could potentially add liquid and create emulsification process within the HME that has not been explored as much for formulation of nanoparticles. So I'm going to go right into the experimental design for H2NAM-loaded lipid nanoformulations, nanoparticles using HME. So if you look at the formulation design, we are again looking at, so what we are doing is first we are melting the lipids that we have, adding the drug in it, melting it, and then feeding that lipid plus drug mixture into the machine. And in the process for emulsification, adding water while the system is in process. So we are running it at a very low temperature, around 70 degrees Celsius, at a very low torque, less than 10%. So the system is very smooth, seamless, and the process for the whole formulation to be made is about 30 to 45 seconds for the batch we are preparing. So this slide shows you about the composition of formulation. You'll see we used multiple components. Everything that we have used is FDA approved, has been used in products. We use labrafil, polysorbate, propylene glycol, polyethylenamine, and the drug, 50 milligram drug per formulation we loaded. The reason for using polyethylenamine is because it provides positive charge on the surface of nanoparticles. You would think, where would it be important for you? Why would you need positive charge going into the lungs? This positive charge, because of polyethylenamine, can be used to create a mucus-penetrative nanoparticle that would be capable of getting through the mucus and killing the infection there. That's number one, right? Second question that you may end up asking over time, why do you want to make nanoparticles for H2NAM? Why wouldn't you just stabilize this drug in the current solution somehow, right? H2NAM is subject to extensive efflux transporter by P-glycoprotein in bacteria. So the bacterial biofilms develop resistance against H2NAM because they're able to throw it out. If I could load it in lipid nanoparticles, they will not be able to, we could bypass this process. So that was one of the reasons. So now if you look at, so we developed 5 formulations, F0 to F4, depending on the amount of polyethylenamine we added, or PEI. So you will see all the formulations showed me an entrapment of about 80%. Or so, and so that means we were able to entrap around 40 milligram drug in each formulation going forward. So now, if you look at this, the properties of this drug, so you will see the black bar in these all 3 graphs on the top represent the formulation as we got it out of the hot melt extruder, and the white bars show when we subjected it to sonication for 3 minutes, probe sonication. So you will see that the formulations that we get in almost all the cases, we got sub 200 nanometer formulations for all 5 trials, right out of the HME. When we sonicated it, we were able to, the PDI was a little bit higher right out of the sonicator, but when we sonicated it, our formulations average size was somewhere between, depending on the concentration of PEI, between 35 to 60 nanometer, which is pretty good for us. Now, for us, because we want to develop inhaled formulation, and inhaled formulation is you have nanoparticles, but you need a device to inhale it. In this case, because it's a liquid formulation, we need to have a nebulizer. So we wanted to see what happens when you take this formulation, subject it to nebulization. So does the formulation retain the drug content, retain the particle size, what essentially happens? So we ran this formulation through a PARI-LC plus nebulizer, one of the most commonly used nebulizers out there. And we saw that particle size was increased a little bit, but in most cases, we were still around 60 nanometers, and the drug content was almost 100% in all the cases. So we did not lose any drug, we did not lose formulation integrity in the process of nebulization. This slide essentially gives you solid state characterization. You can see that with the, and again, we are showing data here only for a couple of formulations because there are 5 of them, so it becomes too much. So with after extrusion, both the formulations became completely amorphous. Some of the cryo-TEM images on the side, you can see the particles clearly here. Now looking at the drug release, the release on the left side shows the typical PBS 7.4 pH 37 degrees Celsius release. You will see that the release is is sustained over a period of time, with us getting about 80% release at around 8 hours. So that would be enough, given the half-life of the drug. It would be enough for us to achieve about once a day delivery. But this PBS, when you look at, and you will also see that as the concentration of PEI changes from F0 to F4, increasing PEI concentration, you see slight little bit of sustained release effect, right? You see less burst release with formulations with more PEI. But that's a release with PBS that has nothing that your lungs have. It does not have all of the surfactants, all of different concentrations of different things. So we ran the release again in simulated lung fluid, which has DPPC built into it as a surfactant, which is naturally present in the lungs. So the graph on the right shows you that, and you see that sustained effect becomes more prominent, because with the positive charge, the polyethylenamine-based formulations start interacting with DPPC. You will see that with formulation F3 and F4, we get a very nice sustained release up to with around 85 to 90% drug release around 8 hours. Again, validating the point that we can develop this as once-a-day formulation. Now, the most important part is you got these nanoparticles, they were great, the release is outstanding, but can they be inhaled? Is the body going to throw them out, right? So for this, we ran an experiment known as cascade impactor, next-gen cascade impactor, to find out a parameter called mass median aerodynamic diameter. So mass median aerodynamic diameter, or MMAD, essentially represents the diameter, how that nanoparticle or the formulation will behave like in terms of deposition across the lungs. It's not the physical size of formulation. Physical size of this formulation is around 50 nanometers, right? But once you inhale it, once you nebulize this formulation, multiple nanoparticles form a globule. When they form a globule, the aerodynamic behavior of that globule will define where and if the nanoparticle are going to get deposited into the lungs, or they are going to get passed on into the blood, or they are going to get expirated by the lungs by expiration. Right. So if you see here, so what typically we are looking for, based on the literature, for efficient deposition into the lungs, we are looking for an MMAD between 2 to 5 micron. Right. So when we run this experiment, we use a true nebulizer used in humans, PARI-LC+. We have multiple other nebulizers as well, which we have been using recently, and we run it through the machine at a given pressure, given vacuum of 15 liter per minute. That represents human lungs inspiration capacity. And then we see at different stages that define different places in the lungs, how much formulation got deposited at each stage. So and then from there we calculate mathematically the MMAD. So if you see here, the MMAD for F1 and F4, and we did this for all 5 formulations, is around 3.7 to 4 micrometer, well within the range of 2 to 5 micron that is essential for efficient deposition into the lungs. So we are right on there. We have around 65% of fine particle fraction. That means 65% of the formulation that was accumulated into the lungs is in the deep lung region, is within the respirable range. So this is pretty good. Anything about 60 is okay with that. Then we tested the stability. We did stability at 40 degrees Celsius and 75% relative humidity. We tested for particle size, drug entrapment, zeta potential, and polydispersity. Did not see any significant difference in any of them after 4 weeks of storage. And from there, so until now we have developed these formulations. We have set up a procedure that is a single-step process, can be used with HME. The formulation is stable, the formulation is respirable, the formulation does not change, maintains its integrity after nebulization. Now, does it actually work, right, against the bacteria? So we ran an in vitro study on Pseudomonas aeruginosa using rhesus urine assay. So rhesus urine assay essentially, so it's a color-based assay. So you will see the pink color, that means bacteria are alive, right? When you see the color turning into slight purple, bluish, that means the bacteria are dying or there was no bacteria. For example, if I add this rhesus urine, if you see the control here, it's all blue, right? That doesn't mean that all bacteria are dead here. It means it's my control there. I did not add any bacteria to make sure my assay is working. So we grew the bacteria on the plate, and then you will see we treated them with increasing concentration of aztreonam, either the plain drug, or we tested two different formulations here, F1 and F4. And in the world of bacterial killing, we look at a concentration called minimum inhibitory concentration, or MIC. That's a concentration, visually speaking, when you start seeing a little bit of blue color in your well. So you will see with aztreonam itself, with free drug AZT, the MIC is around 1 to 2 microgram per ml. When you treat it with AZT-loaded lipid nanoparticles, F1 formulation, you will see we saw around 2 to 3-fold improvement in minimum inhibitory concentration, and it is around 0.5 to 1 microgram per ml. With F4, we saw slightly higher, around 2 microgram per ml minimum inhibitory concentration. So we can see that the formulation, we were able to develop a formulation that's stable, that's scalable, and that works either better or at least the same as the current drug. So that way, we can clearly say that we can take the formulation, instead of 3 times a day dosing, we can move on to once a day dosing using this. We are currently doing some in vivo pharmacokinetic studies and in vivo lung infection studies on this particular project as we move forward with this. So to summarize this part of the talk, and then I have a little bit more, if you have some more patience, I can talk about one more thing we've been doing. So we successfully achieved continuous production of lipid nanoparticles using hot melt extrusion. Again, we didn't use a lot of hot and melt part in here. We mostly use extrusion part in this case because we did not go too high in the temperature. Excellent in vitro aerosolization and antibacterial activity. It's an economic technique to prepare inhaled nanoparticles. I call it economic because literally every pharmaceutical company has the equipment and has the capability to scale it. You do not require to install, prepare, develop new facilities for this. And we see enhanced stability and extended release formulation. So we created a ready-to-use formulation, not ready-to-constitute formulation that is stable under accelerated condition for a month and could be used as is. So now with this, we talked about lung infection. Now I wanted to give you another case study that we recently published with help from Croda Pharma in terms of excipient, preparing inhaled nanochemotherapy by using hot melt extrusion. So again, the similar concept, but in this case, we are looking to treat lung cancer, right? So again, I just keep going back to the same question. If the disease is in a certain body part, why can't we deliver drug to that part? Why do we have to use an oral dosage form? I understand it's patient compliance, or into the veins of the patient, if we can develop systems that could deliver the drug right into where the disease is, decrease the drug concentration. The more of the reasons is, as I keep saying, less than 5% of the drug that is injected into the body reaches to the lungs. Maybe it could even be less, right? So just by the fact of changing the route of administration from injection to inhaled, we could potentially, theoretically reduce the dosing by 20-fold, right? Because that way we'll be able to achieve the same drug concentration in the lungs by going inhaled. Now, but if you go inhaled, so inhaled chemotherapy has been around for a while. It's being tested for a while. However, again, we end up into technical challenges dealing with the formulation development and dealing with use of lungs as a route of administration, right? Many cancer drugs are hydrophobic. Many suffer from low permeation and low bioavailability. Nanocarriers can circumvent all of the challenges, as we recently discussed. Again, scalability is a problem. So we kind of took the similar approach. In this particular formulation, tested 3 different lipids, lipids and surfactants, L18POG, propylene glycol, and twin 80. I'm not going to go delve into too much into the details of it, but the paper, we recently published this study. So we optimized 2 formulations that are 1:1 ratio of L18POG and propylene glycol, E51 and E52. 1 and 2 represents the ratio of water in the formulation. One similar ratio, and 2 times water. So you'll see drug loading for these formulations, and we use erlotinib as one of the most commonly used tyrosine kinase inhibitor drugs. It's very water insoluble, the free base form. We were able to load 4.8, around 5 milligram drug per milliliter of the formulation, which is pretty good for lung delivery. We ran a solid state characterization. You see the drug has been converted into completely amorphous form. You can clearly see formation of lipid nanoparticles. The release is almost zero order. You will see here the inhalation aerosolization properties are excellent with an MMAD of around 3.9 micrometer with fine particle fraction of about 76%. That is great. That means 76% of the dose that gets delivered to the lungs reaches to the lungs and gets deposited. We also ran extensive biological studies. This is just a part of it. So we ran cytotoxicity studies, simple plain MTT assay. We see that just by encapsulating the drug into nanocarriers, we were able to reduce the IC50 for the drug threefold, from 7.7 micromolar to 2.7 micromolar, just by encapsulating. We also ran some 3D spheroid studies, as you will see from the graphs here, and then some microscopic images here. We see that nanocarriers behave significantly better than any of the plain drug formulations. So we are able to reduce the drug dose significantly. So to wrap it up, stable erlotinib-loaded nanoemulsions were prepared using HME. It's a single-step synthesis. The formulated nanoemulsions can be developed as a nebulized therapy significantly, and they can be nebulized for local lung delivery to the therapeutics. Again, we have capability, talking about St. John's. My lab is funded by many pharmaceutical companies. Sometimes we work as a collaborator, sometimes we work as kind of a contract research organization for them, where they have products to develop. We help them develop the product. We have facilities available and ready to go, all the way from formulation optimization, excipient selection, all the way to in vivo studies, preclinical animal model testing as well. Thank you so much for your time. Happy to answer any questions.
Thank you very much, Vivek. Obviously, I know it's been a long day, but any last questions? Yes, Bob.
Two questions. A really nice talk. The last part where you had 3.9 micron particles, which are perfect for aerosolization, how much work did you have to do to get the extrusion to give you that particle size?
It's a single run.
But in terms of surfactant loadings, RPMs,
from what I remember, the RPM for extrusion was about 300 RPM. So because we are running a liquid formulation, so torque is pretty low. We are not going more than 65 to 70 degrees Celsius in temperature.
And then a question earlier, when you were talking about the aerosol delivery for cystic fibrosis, you said you titrated in PEI, makes the surface cationic. Is mucus binding a problem for delivery, you think, because of cationic particles are found in mucus?
Not necessarily, because in most cases we are looking at dealing with biofilms of bacteria, and the mucus is pretty thick, so even if the particles are able to get through the mucus, they should still be okay in terms of delivery. Does that answer your question?
Okay. All right. Thanks, Bob. Any additional quick questions for Vivek before we wrap up? No? All right. Well, thanks again, Vivek, for joining us. Thank you.
Thank you.